This lecture is all about the bacteriology laboratory. We're going to talk about aerobic and anaerobic bacteriology. And we're going to start with the very beginning, and you'll see on the next slide, starting with beginning definitions. So, what's an obligate aerobe? Well, an obligate aerobe requires oxygen to grow. What's an obligate anaerobe? Well, obviously, they don't like oxygen, so if they're exposed for more than about 30 minutes of oxygen, they will not continue to grow. It's toxic and they will die. What's a facultative anaerobe? Well, these are organisms that we mostly deal with. They're aerobic and anaerobic conditions, and they are the organisms like Staphylococcus and Escherichia coli. So really, the organisms we deal with most of the time are facultative anaerobes. What's microaerophilic? Well, they're kind of an unusual group of organisms, mostly Streptococci, and they grow better with reduced oxygen and elevated carbon dioxide. There are aerotolerant anaerobes. I just said anaerobes don't like oxygen, but there are some anaerobes that will grow in the presence of oxygen and will not be killed by prolonged exposure to oxygen. One of the primary examples, and one to remember, is Clostridium tertium. Then we have two definitions of growth of organisms, the lag phase and the log phase. Lag phase is when the organisms are getting kind of old. They're older than 24 hours. They grow slowly, and maybe they're not very appropriate for biochemical or susceptibility testing. Then we have the log phase organisms. They're exponentially growing. They are appropriate for biochemical and susceptibility testing because they're adequately producing enzymes and resistance markers. Then we get into stationary phase. These are appropriate for just the transportation of specimens. They keep the organism viable, but they do not have any growth nutrients to increase the numbers of organisms. On the next slide, we'll go into specimen collection for aerobic bacteriology. First, about throat and wound collection, which is normally using a swab. When you collect a swab from a patient, the swabs will be placed in either Stewart's or Amy's transport media. This is a buffered solution with peptones. These solutions will preserve the viability of the organisms, but they will not promote growth of the bacteria. These swabs are not cotton. Cotton swabs can be toxic to organisms, and organisms can be trapped in the cotton fibers. These swabs are actually made of polyester, or some of the newer swabs are sponge-like material, which allow the organisms to attach but then quickly elute off of the sponge fibers. When we get urine, there's two methods that we usually get urine submitted to the laboratory. One of the most common things done is some kind of suspension into a stationary phase with the use of boric acid. With urine cultures, we're normally doing quantitative assessment, so we do not want the numbers of organisms to increase. So you can use boric acid to put the organisms into a stationary phase during transportation, and then once they're onto solid media, they will continue going about growing their merry way. Another thing you can do with urine is refrigerate it, but it has to be refrigerated after 1 hour, and of course any urine specimen is no good after 24 hours. With refrigeration, same theory applies as with the boric acid. You will stop the organisms from actively growing, and you will maintain the original colony count, but also the viability. On the next slide, we're going to talk about the gram stain procedure. Gram stains procedure has been around for decades, and it remains a very, very good way for us to assess specimens for organisms and cellular material. It's a multi-step staining procedure. Oftentimes it's done by automated instruments now, but let's dissect it a little bit and then maybe you'll understand a little bit more how the gram stain works. First, you have to start off, of course, with a smear of your organism or specimen that you're wanting to assess for morphology and cellularity. You put that specimen and you need to fix it, usually on a heat block. So you let the proteins dissolve a bit and adherence happen to the specimen will adhere to the slide during the fixation process, so you won't wash it all off during this multi-step procedure. After fixation on a heat block for about a minute, you add the crystal violet stain. This is termed your primary stain, stays on for approximately a minute. Rinse off with sterile water, and then you add your iodine treatment. The iodine is a mordant and will force the gram-positive stain to stay in a cell wall that contains peptidoglycan, or a gram-positive cell wall. The next step is the decoloration step, usually performed with acid alcohol. It's 5 to 10 seconds of decolorization, and then you put on your counter stain, which is normally the stain safranin, for a minute or so. At the end of this procedure, you'll have known what's gram-positive cell wall, which is stained a darkish bluish purple color, and a gram-negative cell wall, which will be stained red. On the next slide, we will talk about the appearance of gram stains. If you want a more intense study of gram stain morphology, there is a specific lecture on my blog just about gram stain morphologies. But let's just look at some of the more common ones that we see in the laboratory. On the upper left hand is the Streptococcus. Notice they're gram-positive cocci, they're blue in color, but they're chaining in long chains or ropes. Underneath that are the appearance of just a regular enteric gram-negative rod like an E. coli, boxcar-shaped, red in color. It's a gram-negative rod. Below that is a gram-positive rod. It's a bacillus. It's blue in color, boxcar-shaped. Upper right, one of the more classic gram stains in micro is the Fusobacterium. Fusobacterium are fusiform-shaped, very faintly staining gram-negative rods. Very characteristic good gram stain to remember. And then on the lower right is your classic gram-positive cocci in clusters associated with the Staphylococcus. On the next slide, we talk about how a gram stain can be used to judge a sputum specimen. Judging of a sputum specimen has been around for decades and is very important for determining if you actually received a specimen of sputum from a patient or did they just spit in the cup. And so we don't want to culture just spit in the cup. So this is the way we can tell in the micro lab if it's a good sputum and we should continue with our culture. We take the specimen and we do a gram stain. We then look at the gram stain under a low power magnification on a light microscope and look for epithelial cells. If you look at the bottom left, you can see that that slide is full of epithelial cells. That would be judged a bad sputum or a spit. We would then reject that for bacterial culture and do the proper notification and ask for a new sputum. Hopefully the new sputum will actually be a deep cough specimen, as pictured on the bottom right. You can see the bottom right picture does not have any epithelial cells and it's full of polys. So that is a good specimen to continue on in the culture process. On the next slide, we're going to start talking about the most common agars that are used in the bacteriology laboratory. The most common agar that we use is a 5% sheep's blood agar plate. The blood agar plate is important for the beginning of the workup and morphology of many of our organisms because we can gauge the hemolytic reaction on the blood agar. If you look at the arrow pointing on the left-hand plate, you will see that there's alpha, beta, and gamma hemolysis. Alpha is the greening of the agar. Beta is the complete clearing of the agar, and gamma has no hemolysis. Blood agar plate is very good for the growth of your common gram-positive and gram-negative bacteria. It'll also grow yeast. Now the plate on the right-hand side is chocolate agar, and you can see that it looks different. It's a brownish in color like chocolate. It does not taste like chocolate, but it does look like chocolate. Chocolate is actually caramelized blood agar. The blood is added to the agar when it comes out of the autoclave when it is still quite hot. So the blood actually cooks and caramelizes, making it this brown color. It is then filtered and poured into the agar plates. This media is more for fastidious organisms and is more enriched than your regular blood agar plate. It will grow organisms such as Haemophilus influenza and Neisseria gonorrhoeae that your blood agar plate will not grow. On the next slide, we talk about the agars which are most used for gram-negative rods. McConkey agar is the one that's most commonly used, and it is termed a selective and differential agar because it selects for the growth of gram-negative rods by the inclusion of crystal violet, which inhibits the gram-positive organisms. And it also is differential because you can determine lactose fermentation on this agar. As shown in the slide, you can see on the left-hand side where the positive is, that is the color of lactose fermentation. Lactose fermentation turns this bright pink color due to the neutral red indicator. On the right-hand side of that plate, you can see that there's no color produced, and that is a non-lactose fermenter. After you've put your patient specimens on either your blood, chocolate, or McConkey agar, of course, then you need to incubate them. The standard incubation temperature used in bacteriology laboratories is 35 degrees centigrade. We can either use room air incubators or those with increased CO2, and you normally incubate these agar plates 24 to 72 hours. On the next slide, I give just a very, very brief explanation of what is MALDI-TOF. MALDI-TOF mass spec stands for Matrix Assisted Laser Desorption Ionization. That's a very, very long name for what we're really doing is instead of using biochemicals like in the old days, MALDI-TOF is a step into the future. And what we're doing is we're identifying organisms by analyzing their proteins. If you would like to learn more about this method, there is a lecture on this blog specifically talking about MALDI-TOF organism identification. This is really what some of the major, bigger laboratories are moving toward, but most of the rest of our lecture, we're going to be talking more about the standard older biochemical methods, which are still very, very important and very important for the study, particularly for board exams and such. We'll move on to the next slide and start talking about some of the individual organisms. And we're going to start talking about the gram-positive cocci, the Staph, the Strep, and the Enterococcus. On the next slide, we start with the Staphylococcus. You will look on these gram stain slides on the left and right hand side, and you will see that these organisms are in gram-positive cocci in clusters. And we'll learn to identify these Staphylococcus using this flow chart that's on this slide. We saw the gram-positive cocci in clusters, but the next step is, do they have clumping factor? And they were divided in two groups based on clumping factor. Also, all of the Staphylococcus are positive for the enzyme catalase. We follow our flow chart down. You see on the left-hand side Staph aureus. Staph aureus has the coag enzyme that is positive. As the name implies, coagulase negative staph do not. They do not have the coag enzyme. Staph aureus are also kind of yellow in color and slightly beta hemolytic. Normally, the coag negative staph are white in color and not hemolytic at all. If we go to the next slide, we can see what really is the catalase enzyme reaction. This is really a very, very easy old school reaction to do. It's using hydrogen peroxide is put onto the microscope slide, and you put a little bit from a wooden stick of organism into the hydrogen peroxide. You can see on the left-hand side there are no bubbles, and on the right-hand side it's full of bubbles. That's how easy this test is. So the negative is no bubbles, the positive has bubbles. This determines whether an organism has catalase enzyme reaction. You can see on the right-hand side is how the Staphylococcus would react. They are all catalase enzyme positive. If you go to the next slide, you can see the tube coagulase reaction. This is also a very simple old-style test, but is also very important in the history and the identification of Staphylococcus. This test is done by putting a small amount of rabbit plasma in a test tube and inoculate it with your suspect organism. You read it at both 4 hours and 24 hours after incubation at 35 degrees centigrade. After 4 hours, if you have a firm clot in the tube, like shown in the bottom of the picture, that shows that you have clumping factor, and that's Staph aureus. If you look at the upper tube, there is no clot at all. At 4 hours, if there is no clot, you have to re-incubate for 24 hours. After 24 hours, you still have no clot, then that is a coagulase negative staph. The one little trick with this test and how it can get you into trouble, if you don't read it at 4 hours, you go, oh, I'll just read it at 24 hours. Well, that can be a problem because sometimes you can have a clot formed at 4 hours, but it will disintegrate by 24. So you could actually miss a Staph aureus if you don't read at both 4 and 24 hours. On the next slide, you will see how we've moved forward and away from the tube coagulase test. There's nothing wrong with the tube coagulase test, it's just slow, and things aren't slow in microbiology anymore. So we've moved on to doing rapid slide agglutination tests for the identification of Staph aureus. This slide outlines one of them known as a Staphylococcus test, where you have a latex agglutination particle that's bound with protein A and coagulase. And this is found, the protein A and coagulase are found on the surface of the Staph aureus, and your latex bead is coated with fibrinogen and immunoglobulin to protein A. So you can actually do a slide agglutination test to determine if your organism is a Staph aureus or not. You can see on the black slide, the upper right left-hand corner shows clumping. That would be the presence of the latex particle with Staph aureus. Below it looks like milkish colony, or I should say milkish solution. That's no clumping, and that's what happens when you put a coag negative staph in one of these reactions. The advantage of using this type of methodology over the tube coagulase is you can actually get a reaction in about 30 seconds rather than the 24 hours, which you get with a tube coag test. On the next slide, we talk about the virulence mechanisms of Staph aureus. The primary virulence mechanism is protein A. This is a surface protein and has the ability to bind immunoglobulin and combat the immune response. Staph aureus is a very potent pathogen, and there's many diseases and associated toxins of Staph aureus. We start off with toxic shock syndrome. It has the TSST1 toxin. The scalded skin syndrome, known for the exfoliating toxin. Notice the picture which the arrow is pointing to? That's what happens in scalded skin syndrome. This unfortunately can happen in neonates. It can cause onion peeling of the skin. In the early 2000s, it was very common in the literature and in our society. We had a lot of soft tissue infections being caused by Staph aureus. There is a slide there on the lower right-hand side of a lesion, a soft tissue skin infection with Staph aureus or MRSA caused by the PVL toxin, Panton-Valentine leukocidin. Staph aureus is the primary cause of septic arthritis. It also has a potent enterotoxin that is heat stable, that is capable of causing food poisoning. It's primarily a vomiting type syndrome. And then it, of course, is a pathogen of bacteremia and endocarditis. All of these toxins act as superantigens. They recruit host defense cells, and they liberate cytokines with systemic effects. On the next slide, we talk about the primary resistance issue of Staph aureus, and that's Methicillin Resistant Staph aureus, or more commonly, we just call it MRSA. MRSA, of course, stands for Methicillin Resistant Staph aureus and is due to altered penicillin binding proteins, or PBP2. These are produced by the MECA gene and codes for resistance to oxacillin, methicillin, and nafcillin. All our MRSAs are considered resistant to semisynthetic penicillins and cephalosporins. You do not need to do laboratory testing to prove this. It's by definition. This makes vancomycin one of the drugs of choice. There has been creeping resistance, or creeping increase, I should say, in MIC values of vancomycin for Staph aureus, but vancomycin still is often the drug of choice for MRSA. One of the things in the laboratory that has changed over the last decade is we used to rely on oxacillin to determine resistance and the definition of methicillin-resistant Staph aureus. However, over time, researchers have shown that sulfoxetine is a better way to determine MRSA in the laboratory than by using oxacillin. So you can see on the bottom right-hand side a Kirby-Bauer disc method with a sulfoxetine disc. And this is showing growth right up to the sulfoxetine disc, which would be what happens with MRSA. On the next slide, we talk about the D-test. The D-test is really an interesting test, so called by the determination of the zone size looks like a D, and this is to determine if Staph aureus or MRSA are susceptible to clindamycin. During therapy, Staph aureus isolates resistant to erythromycin possess enzymes capable of inducing clindamycin to become resistant, so therefore clindamycin wouldn't be useful for therapy. Clindamycin is often used as a topical agent for soft tissue infections and such, and for people who are allergic to many antibiotics, clindamycin can be used for systemic infections. So it's very, very important that you have an accurate judge of whether clindamycin is susceptible or not. To do the D-test, we simply use a blood agar plate and place clindamycin and erythromycin discs at a suitable separation point on a lawn of the Staph aureus that is to be tested. We incubate it for approximately 24 hours and we look for the zone sizes and how they appear on the agar. If you look at the bottom right hand picture, you can see that the D-test is negative and the zones are round and nicely formed. But if you notice the picture on the left, you can see that the clindamycin disc has been blunted by the effects of the erythromycin being resistant. And instead of having a nice susceptible round zone of inhibition, it's in the shape of a D, so-called D-test. This is because it is being induced to become resistant, and that's why that side of the zone is blunted and you have a deformation. Inducibly resistant Staph aureus isolates with clindamycin should be reported as such, and clindamycin should not be used as therapy. Let's move on to the next slide. This addresses Staph aureus or MRSA as hospital epidemiology issues and talks about nasal surveillance for MRSA and Staph aureus. Nares is your most common colonization site for these organisms, and they can be a source of transmission to others and to a patient's own wound sites. So it's very common for hospitals to do surveillance testing on incoming patients. The most common thing that is done is to use what's called chrome agar. Chrome agars are selective and differential media that contain chromogenic substrates, and they will turn specific colors if Staph aureus is grown on the media. So there's two types of media shown in this picture. One company's media, Staph aureus, turns blue, and the other, Staph aureus, turns a mauve or pink color. You can also add sulfoxetine to these plates to detect MRSA. So if the plate contains sulfoxetine, you're looking for MRSA. If they do not have sulfoxetine, it becomes a very nice screen plate for just Staph aureus. These results are usually available in 24 hours. If you need a more rapid screen, albeit more expensive, you can use molecular assays. They can also screen for Staph aureus and MRSA and are slightly more sensitive than are the chrome agars. If you do detect Staph aureus or MRSA in nasal and it needs to be eliminated, mupirocin is normally susceptible and most often used for ridding people of nasal colonization. On the next slide, we talk about the coagulase negative staph. We often see them as just normal skin contaminants or contaminants in culture, but some of them are quite capable of causing infection. Let's first talk about Staph epidermidis. It is the most common coag-negative staph that we see in the laboratory. It's the major component of the normal skin flora. It's a common cause of subacute bacterial endocarditis, however, usually from the mouth, from dental procedures. Pathogenicity of the Staph epidermidis is from its cell adhesion factors and its ability to form biofilms on biologics and plastics. So this is a particular issue if you have catheters in place or replacement joints or even prosthetic heart valves. So it can, and when the coag-negative staph is encased in these biofilms, it can be very, very difficult to treat. Second important coag-negative staph is Staph saprophyticus. This is a cause of urinary tract infections in the childbearing age female. This species of coag-negative staph adheres in greater numbers to the epithelial cells in these areas. The way we can screen for Staph saprophyticus is by using the novobiocin disc. If you see the picture on the right-hand side, you can see that the organism grew right up next to the novobiocin disc. This would be the characteristic reaction of Staph saprophyticus in a novobiocin-Kirby-Bauer reaction. It is the only coag-negative staph which is resistant to novobiocin. If you go to the next slide, we talk about Staph hemolyticus. Staph hemolyticus can cause sepsis and can be a pathogen in blood cultures. This is the only coag-negative staph that is hemolytic on blood agar, so be careful. Don't confuse it with Staph aureus. Staph aureus is almost always beta hemolytic, as is Staph hemolyticus. But of course, Staph hemolyticus is coagulase negative. The next important coag-negative staph is Staph lugdunensis. It is positive in the PYR test and will turn pink as it pictured in this test. It will turn pink on the PYR disc. Staph lugdunensis is thought to be important because it is perhaps more pathogenic than your other coag-negative staph. And finally, let's talk about Micrococcus. No, Micrococcus is not a coag-negative staph, but oftentimes it can be confused with one. But however, it is a bit different in that it produces kind of this obnoxious-looking yellow color on the plate, but it is catalase positive. But look at the gram stain. If you notice, the gram stain's in tetrads, and that is different than the randomly clustering of the Staphylococcus. It's tube coag negative. It does not ferment glucose, as does some of the other staph species. And it is primarily an environmental contaminant and is not usually found in human infections. I say almost never. It's almost never. Therefore, we don't want to confuse it with organisms that might be treated with antibiotics. The micrococcus should not be treated because they are almost always environmental contaminants. Let's move on to the next slide, and we're going to start talking about the Streptococcus. The Streptococcus are gram-positive cocci in chains and pairs. If you observe the gram stain, you can see they are very, very elegant-looking chains of gram-positive organisms. On the left-hand side, you can also observe the hemolytic reactions on the blood agar plate. These organisms are all catalase enzyme negative, and that helps us differentiate them from the staph, because remember, the staph are all catalase enzyme positive. Also, the Streptococcus are grouped by the hemolytic pattern on the 5% sheep's blood agar plate. We have alpha, beta, and gamma hemolysis. Alpha hemolysis is greening of the agar with partial hemolysis of the RBCs. This includes the organisms Viridans Strep, Strep pneumonia, Granula catella, and abiotrophia. The B on the plate is for beta hemolysis. This is clearing of the agar, and as you can see all the way through the plate. It's complete hemolysis of the RBCs, and this is what we see most characteristic with the beta hemolytic Streptococcus. And then finally, the bottom one is the gamma symbol, and this is for no clearing of the agar or gamma hemolysis. The red blood cells are intact. This is such as the organism Streptococcus bovis. On the next slide, we'll start talking about the beta hemolytic strep. The beta streptococcus are grouped using the C carbohydrate in the cell wall of the bacteria. This carbohydrate helps us group the beta hemolytic strep into the most common human infections of A, B, C, F, and G. These are our most common beta hemolytic strep. The way we can identify these is by using a very old system called the Lancefield grouping system. This uses the C carbohydrate in the cell wall of the beta hemolytic organism. We do an extraction and do a slide agglutination reaction with purchased monoclonal antibody. This is attached to latex beads and causes agglutination, as you can see in the slide in the bottom. This immunologic typing system is the best system available for us to identify the beta hemolytic strep, and is far better than the use of biochemical reactions. If we go to the next slide, we will talk about Strep pyogenes. Strep pyogenes is the genus and species name of group A beta hemolytic strep. If we want to use biochemical reactions to identify the beta strep A, Strep pyogenes, the two most common ones are bacitracin-Kirby-Bauer testing and the PYR reaction. In the bacitracin-Kirby-Bauer, you basically just do a sensitivity test using the bacitracin disc, as you see in the picture on the right-hand side. The bacitracin disc usually has the letter A on the disc, and you can see that there's a zone of inhibition around this disc. This would be considered a beta hemolytic strep group A and sensitive to bacitracin. However, we do have to be careful because the bacitracin disc is not just inhibitory for group A, it can also be inhibitory for group C. The PYR biochemical test can also be used to identify group A strep. Yes, we did just talk about PYR with Staph luginensis, but it can also be used with the Strep pyogenes as an identification test. We would know that the organism is beta hemolytic and catalase negative, and then perform the PYR test. The way this test is performed is you have the paper disc in which the PYR reagent is on the disc. You moisten the disc and add your organism, and then incubate for 2 minutes at room temperature and add a cinnamaldehyde reagent, and you look for the color reaction. The pink color is positive in the PYR test, pink for positive, and the Strep pyogenes will turn pink on the PYR disc. As I said, this disc is not just for beta hemolytic strep group A. There are other organisms, namely Staph luginensis, and we're going to talk about the Enterococcus, and they're positive on the PYR disc as well. One of the things that I found increasingly interesting about Strep pyogenes is its ability to maintain its susceptibility to penicillin. In all this age of antimicrobial resistance, Strep pyogenes is still sensitive to penicillin and cephalosporin antibiotics, and they are the therapy of choice. On the next slide, we talk about the virulence mechanisms of Strep pyogenes. The primary mechanism of virulence is the M protein, and it prevents phagocytosis. Also, there is a capsule. It's a hyaluronic capsule that protects also from phagocytosis. One of the really interesting little things about Strep pyogenes is its ability to produce streptolysin O and streptolysin S toxins. If you look at that plate that I have pictured, you can see that there are some more intense areas of hemolysis than others. How this all works is streptolysin O is oxygen labile, and streptolysin S is oxygen stable. If you have a group A strep streaked onto this agar plate and you stab into the agar, the areas with the more intense hemolysis are areas where both the O toxin and S toxin is being demonstrated. The areas of less toxin or less hemolysis are where only the S toxin is being demonstrated. This streptolysin O and streptolysin toxin formation by the beta strep group A also comprises the ASO titer, which is important in many diseases. On the next slide, we talk about the strep pyogenes diseases. There are many, and they're very, very bad diseases. We start off with pharyngitis, impetigo, shown in picture number 1, erysipelas, another skin type infection, in picture 2. Very serious cellulitis can be caused by strep pyogenes, shown in picture 3. Necrotizing fasciitis in 4. There can also be puerperal sepsis, classically is associated with strep pyogenes. Toxic shock syndrome can be caused by strep pyogenes, and scarlet fever, which is pictured in picture 5. On the next slide is discussed the very important sequelae of strep pyogenes infection. There's 2 important sequelae, rheumatic fever and glomerulonephritis. Rheumatic fever is primarily caused by inadequately treated strep throat. There was a rise in 1990s of rheumatic fever because unfortunately we were misusing the EIA paper membrane tests to detect children with beta strep A sore throat. Instead of backing up with culture and confirming the disease of strep A, a lot of physicians determined therapy on the positivity of the EIA reaction. The enzyme immunoassays were only about 60 to 70% sensitive, so some of the beta strep A pharyngitis was not being treated adequately, and this was thought to be one of the issues with the rise in rheumatic fever during the 1990s. There is a similarity between the proteins of strep A and muscle tissue causing the immune system confusion, which leads to immune system to attack the heart, joint, and bones, in the sequelae of strep A known as rheumatic fever. The anti-streptolysin O can measure the amount of antibodies present and help in the diagnosis. In glomerulonephritis, this is usually 10 to 14 days following an adequately treated skin infection or pharyngitis. It is, of course, a renal disease with inflammation of the glomeruli. ASO titers will also be positive in this infection, and usually it will resolve with therapy. On the next slide, we talk about Streptococcus agalactiae, which is the genus and species name of group B strep. This has the group B strep as demonstrated in the Lancefield carbohydrate test. If you want to do biochemical testing for group B strep, you could do either the CAMP test or the rapid hippurate hydrolysis test. The CAMP test is as old a test as is possible in the microbiology laboratory. It is performed using a Staph aureus strain with CAMP factor. It is shown in the upper right-hand picture. You streak the Staph aureus with CAMP factor across the horizon and have perpendicular streaks of your group B strep. You incubate it for 24 hours, and then you will see how as the group B strep impinges upon the Staph aureus streak, you get intensification of the hemolysis and the arrow being produced. It's really one of those classic reactions in bacteriology. The second one is the rapid hippurate hydrolysis test pictured on the bottom left. This is a 4-hour biochemical test which can identify group B strep. You put a hippurate disc in saline and introduce group B strep into the saline. You then incubate it for 4 hours and then add ninhydrin reagent. The ninhydrin reagent will turn purple or positive if it is a group B strep. On the next slide are discussed diseases of strep agalactiae. Strep agalactiae is really a pathogen of the 2 age extremes. It is a pathogen of the elderly, mostly causing bacteremia and urinary tract infection, and a pathogen of the neonate, when you have in utero or perinatal organism acquisition during the birthing process. You can have early onset group B strep disease within 7 days or late that can go up to 28 days after the birth process. Treatment is either penicillin or cephalosporins. Pregnant women need to be screened, of course, for the carriage of group B strep, since this is such an important pathogen in the neonate. Screening is done in the cervical and rectal area, and all pregnant women are screened at 35 to 37 weeks of gestation. Enrichment methods for group B strep is a standard of practice. You cannot just simply put a swab onto the surface of a blood agar plate. The standard enrichment mediums are the LIM broth, LIM broth, which you incubate for 18 hours at 35 degrees centigrade. Then you plate the LIM broth onto blood agar to look for your group B strep. You can also use the LIM broth as an enrichment broth for use in molecular testing methods. The second type of broth that can be used is called the carrot enrichment broth. This will turn orange if group B strep grows in the carrot broth. Most laboratories will also go on with this testing and determine if the group B strep is susceptible to clindamycin or not. So this is a very important step for prophylaxis at the birthing time. As you can see on the slide, this one is no longer called Strep bovis. Really, if we properly name it, it should be Strep gallolyticus. And we're dealing with a lot of taxonomy updates in microbiology right now, and this is one of the most important ones because this is a very important organism. We've known it for bovis years, for bovis biotype 1 and bovis biotype 2, but now we're supposed to refer to it as Strep gallolyticus, subspecies gallolyticus, and subspecies pasteurianus. The Gallolyticus subspecies is the one that we used to think of in adults being associated with bacteremia and colon cancer, and that's a very, very important association. The second one was more associated with neonatal meningitis. Both of the Gallolyticus species have the same biochemical reactions. The bile esculin slant, which is shown in the picture with the plus sign, it turns black. This is a precipitin product onto the bile slant. That's a positive bile esculin slant. And then there is no growth in the 6.5% salt broth. The PYR reaction is negative. These reactions are important when we talk about the next organism, Enterococcus, because sometimes these organisms can appear the same on agar plates, and biochemicals are the way that we differentiate them. Strep gallolyticus is susceptible to penicillin, and that has not changed even though we're seeing so much more drug resistance. On the next slide, we will talk about the Enterococcus. There's two common species of Enterococcus, Enterococcus faecium and Enterococcus faecalis. There's no well-defined virulence factors for the Enterococcus. They possess the group D carbohydrate in the cell wall. The biochemical reactions, we need to remember they are important. The bile esculin is positive. You get the growth in black precipitin like we just talked about. But these organisms grow in the 6.5% salt, as shown in the picture on the upper right-hand side. And they are PYR positive, as are group A beta strep and Staph ludenensis. This is important these biochemical reactions are important to keep in differentiation with Strep gallolyticus, which shares the bile esculin positive slant, but the salt negative and PYR negative. And that's the difference with it and Enterococcus. The two species of Faecium faecalis, if you're into biochemical testing, arabinose fermentation can differentiate the two, with Faecium being positive and faecalis is negative. On the next slide, we'll further talk more about the Enterococcus. They can cause a variety of infections. They're a pathogen of opportunity, primarily being found as intestinal normal flora. When they do cause disease, they normally cause urinary tract infection, bacteremia, and abdominal infections. It's important to remember concerning the antimicrobial therapy that there is natural resistance to cephalosporin antibiotics, We should not test these in the laboratory. There's no use. By definition, they are resistant. Ampicillin plus an aminoglycoside, such as gentamicin, can be synergistic for therapy and is used often in cases of endocarditis. There are some unique susceptibility issues associated with the Enterococcus. Primarily, it is acquired resistance to vancomycin, which we know in the laboratory as vancomycin-resistant Enterococcus, or VRE. There's two genes associated with vancomycin-resistant Enterococcus. If you have an Enterococcus faecium, that will be the VAN-A resistance gene. For Enterococcus faecalis, it'll be the VANC-B resistance gene. On the next slide, we will start talking about the alpha hemolytic Streptococcus. It's primarily the main pathogen of this group is Streptococcus pneumonia. Look on the upper right-hand side of this slide. You can see the gram stain of strep pneumo. It's described as bullet gram-positive shaped cocci or lancet-shaped cocci, and they normally appear in pairs with the flattened side up against each other. Oftentimes, you'll be able to visualize the polysaccharide capsule in the gram stain prep. This is the primary virulence factor of strep pneumo and is antiphagocytic. Identification of strep pneumo is all about the biosolubility reaction and optotion sensitivity. The biosoluble reaction, if you look at the plate on the left, on the bottom picture on the left-hand side, you'll see that this plate has had bile solution or sodium deoxycholate placed on top of the colonies. The colonies actually dissolve after about 15 minutes if you're dealing with strep pneumo, hence the name bile soluble. On the right-hand side below is optotion sensitivity. Optotion is ethyl hydrocuprine hydrochloride. It is a biochemical sensitivity, not an antibiotic or any other thing. It is a chemical sensitivity test. That P-disc, which stands for pneumococcus, contains this optotion chemical, and you test for whether it has a zone of inhibition or not. If there's no zone of inhibition, you have an alpha hemolytic strep. If you have a zone of inhibition greater than or equal to 14 millimeters, that is, by definition, a Streptococcus pneumonia. So that leaves us with the biochemical reactions that strep pneumo is bile soluble and sensitive to optotion. If you go to the next slide, we will continue to talk about strep pneumo and how it causes upper and lower respiratory tract infections, sepsis, and meningitis. There is a vaccine available, which is used in young children and elderly, and it is a multi-serotype vaccine and can help prevent invasive infections. There are some unusual susceptibility issues associated with Streptococcus pneumonia. They can acquire resistance to penicillin due to penicillin binding proteins. In this acquisition, you will have an increase in the minimum inhibitory concentration of strep pneumo to penicillin. You have to be careful the way you test strep pneumo. You cannot use Kirby power testing, and you have to use some type of MIC determination to test for whether penicillin is susceptible or not. If you're testing CSF isolates for strep pneumo, the penicillin MIC must be greater than or 2 to be considered resistant. Therapy is, if susceptible to the first-line drugs such as penicillin, that would be your therapy of choice, or possibly a third-generation cephalosporin. On the next slide, we will talk about Viridans Streptococcus. Several species of the alpha hemolytic strep can be grouped into what's called the Viridans strep, and these are found as normal flora in the mouth and upper respiratory tract. Primary species you can see are mutans, salivarius, sanguis, and mitis. The bile esculin slant will be negative for all of these. They are not bile soluble, so that can help differentiate them from strep pneumo, as well as the optotion is resistant with the zone size of less than or equal to 13 millimeters. One of the important things about the Viridans strep is they're a primary cause of subacute endocarditis. And this is also native valve endocarditis as well. And this is primarily causes endocarditis from invasive dental procedures. They can cause abscess and various infections throughout the body, but primarily in the immune-suppressed host. They can have variable susceptibility patterns, and if you do need to treat one of these in an infection, it is best to do MIC testing for penicillin and the other drugs to determine susceptibility. On the next slide, we're going to talk about some of the unique species within the group Viridans strep. This is the so-called Streptococcus aeruginosus group, which include the species Aeruginosus, constellatus, and intermedius. These are found as normal flora in the human mouth, but they're thought to be a bit more virulent than your run-of-the-mill species of Viridans strep, and this is perhaps due to a capsule that they have acquired. They can cause deep tissue abscesses, bacteremias, endocarditis, and intra-abdominal infections, and they can have variable susceptibilities, so it's really best to do susceptibility testing. On occasion, these species can also be microaerophilic, which we talked about with our beginning definitions. These organisms sometimes will not grow in an aerobic atmosphere and do require incubation in the CO2 incubator to be able to grow. The next slide is about the nutritionally variant strep. This is really a unique group of the alpha hemolytic strep. These are vitamin B6 or pyridoxal dependent organisms. So they will not grow on your regular blood agar or chocolate agar medium unless there's additional B6 supplementation. One of the ways that we spot these organisms is they commonly are most commonly found in cases of endocarditis. Far small percentage of overall cases, but this is where we most often see these organisms. The way we can spot them is we will have a positive blood culture, which gram stains for an organism, gram-positive cocci in short chains and such, and we think it looks like a Viridans strep. But when we subculture it onto our blood and chocolate agars, nothing will grow. So what we can do is we can go back, and as the picture on the bottom right shows you, we can do what's called a Staph streak. You subculture your blood all over the blood agar plate, and then you run a streak of Staph aureus down the center of your plate, and then incubate this in the incubator for 24 hours. When you observe the plate after 24 hours of growth, you can see satelliting very small colonies just growing along the strip of the Staph aureus. The Staph aureus supplies the organism the vitamin B6, and these satelliting colonies are your nutritionally variant strep. So this is a way to be able to find this organism. These organisms have been worked on taxonomically over the last few years, and they are now put into two species, the Abiotrophia and the Granulocatella. They most commonly cause endocarditis. They can be more destructive to a heart valve than your normal Viridans strep, so it can be very important to be able to spot these organisms and identify them properly. Sometimes they also will have higher MICs to penicillin and require more therapy than your run-of-the-mill Viridans strep. That's the end of the Streptococcus, and we're going to move into our next group of organisms, which are the gram-negative cocci. And we will primarily be talking about Neisseria and Moraxella. The next slide, we will start talking about the Neisseria species and the Moraxella species. These are very small kidney bean shaped cocci that stain red because, yes, they are gram-negative. They also share the oxidase enzyme reaction. Both of them are oxidase positive. It has been difficult over the years to do biochemical reactions for the Neisseria and the Moraxella because they require some special growth requirements in the performance of biochemical testing. Over time, we've used what's called cysteine tryptase agar carbohydrate fermentations. These are specifically manufactured and supplemented carbohydrate tubes to help us determine the reactions of the Neisseria and the Moraxella. The classic sugars that are tested for these organisms are glucose, maltose, lactose, and sucrose. And you can see the characteristic fermentation patterns on this slide. Notice that gonorrhoeae is glucose, G. Meningitidis is glucose plus maltose, M. Lactamica is normally usual normal flora and not a pathogen. That's why the lactose is included, so you will not get confused and misidentify a Neisseria meningitidis. Neisseria catarrhalis does not fermentate any of these sugars, and it has the positive DNA test. The other things that are different about these organisms is Neisseria gonorrhoeae will not grow on 5% sheep blood agar plate, but Neisseria meningitidis will, as will Lactamica and catarrhalis. Those three will grow on blood agar. Gonorrhoeae is the lone wolf and will not, and will require chocolate for growth. On the next slide, we talk about the carbohydrate fermentation reactions of these organisms. The top picture is of a miniaturized carbohydrate test strip that can test for the sugar reactions of the Moraxella and the Neisseria. You can see that if you read the sugars on top, names of the sugars on top of the strip, and you can see that there's a control well, and then glucose, maltose, lactose, and sucrose. The control well will be the judge of whether a reaction is positive or not. In this strip, the control is positive, and so you can see that you compare the positive to the negative reactions. So on this test strip, you would see that the control is of red color and that the glucose is a positive yellow color. So you compare it to the control, it is more yellow, and so this one would be determined to be glucose positive, therefore Neisseria gonorrhoeae. Please notice that you can also do this test on full-size tubes, and it will turn slightly yellow at the top of the tube. The other thing that's important about these Neisseria and Moraxellas are the oxidase enzyme spot test. These organisms are all oxidase positive and will form a bluish color when exposed to the cytochrome oxidase reagent. On the next slide is a discussion of Neisseria meningitidis. This is a horrible cause of meningitis in young children and adults. The hallmark is petechiae, as you can see in the picture of this small child. The organisms will crowd into the capillaries and cause tissue necrosis. It can also cause disseminated intravascular coagulation due to the endotoxin formation. The infection can be rapidly take over a patient extremely rapidly and be rapidly fatal. Normal carriage of Neisseria meningitidis is approximately 10 to 20% in the general population, can be a tad bit higher in some select groups, and the carriage is in the nasopharynx area, and that's where you would look for carriage. This organism is susceptible to penicillin. Capsular polysaccharide is the primary virulence factor. There is a chronic type of disease where it's not quite as rapidly fatal. This form of the disease is associated with complement deficiencies in the 7, 8, and 9 regions of the cascade. When you look at the anatomic pathology findings of adrenal glands, you can see adrenal necrosis, and this is referred to as the Waterhouse-Friderichsen syndrome. The current epidemic strain in the United States is type C Neisseria meningitidis, and it is the primary problem that has been reported in dorm rooms in college freshmen and moving around the young adult population. There are immunizations available. It does include the type C strain, which in the past has been endemic, but does not include the extremely virulent type B strain. There are new vaccines in development, and hopefully before too long we will have a type B vaccine as well. The next slide discusses Neisseria gonorrhoeae. It's very different than meningitidis in that it causes an acute urethritis, endocervical, ocular, rectal, oropharynx, and septic arthritis. In about 10 to 20% of females with Neisseria gonorrhoeae infection, it will ascend to PID, but very few will really disseminate into the bloodstream. The gram stains of urethral discharge can be very useful for the diagnosis of males, but most people would not consider use of the gram stain in females. It's just not specific enough. It can be normal flora, which can look very, very much like GC in the female genital tract. So gram stains are usually not performed on females. The transport for a Neisseria gonorrhoeae culture has to be specific. We usually use charcoal swabs, and we do not refrigerate any specimens in which we want to isolate Neisseria gonorrhoeae. There's selective media that can be used for its culture because it is so fastidious and requires nutrition. We can use selective Thayer-Martin agar or Martin-Lewis agar. Most people do not do cultures for Neisseria gonorrhoeae anymore. Molecular and amplification methods have really taken over for the detection of this pathogen. It is more rapid, it increases its sensitivity, and is more specific sometimes, and is very superior to culture. So really, amplification methods is your standard of practice and primary method for looking for Neisseria gonorrhoeae in urogenital specimens. GC is known that it can produce a beta-lactamase enzyme, of course, and render it resistant to penicillin. We haven't seen penicillin susceptibility for GC in the United States for probably 30 to 40 years. And also, there's chromosomal resistance. Therapy for GC currently consists of ceftriaxone plus erythromycin or doxycycline, and the combination therapy is to prevent resistance. On the next slide, we can see gram stains of Neisseria species and Acinetobacter species. On the upper left-hand side is your classic intracellular and a few extracellular Diplococcus that can be seen in Neisseria gonorrhoeae and Neisseria meningitidis. On the lower right-hand side, you can see the gram stain of Acinetobacter species. This organism can commonly be found in the female genital tract, and it is one of the reasons why we do not want to read female gram stains for GC, because the presence of this normal flora organism could confuse a morphologist, and you could falsely call a gram stain positive when it is not. On the next slide, I include, it is about Moraxella catarrhalis. This is a cause of pneumonia, ocular infection, sinusitis, and otitis media. The gram stain of the sputum, which is shown on the right-hand side, can be very helpful in the diagnosis of Moraxella pneumonia. You will see many polys with gram-negative intra- and extracellular diplococci. Neisseria gonorrhoeae and Neisseria meningitidis do not cause pneumonia, so when you see a gram stain such as this, your first thought should be to the cause of Moraxella catarrhalis. One of the things that's really unique about this organism is it's described as having a hockey puck colony. And that is if you get a little wooden stick, like your little hockey stick there, and you push this colony around, it will actually push across the agar plate. It will not just gamush up against the loop. It will actually push around like a hockey puck. It is oxidase enzyme positive, DNase enzyme positive, and resistant to ampicillin by beta-lactamase enzyme production, or the so-called cephinase test can be used to test for resistance. The therapy is usually augmentin or a second or third generation cephalosporin. On the next slide, we're going to start talking about the gram-positive rods.